What you see here is a video capture of fluorescence used to measure the thickness of a person's tear film. The darkening patches indicate a thinning tear film that causes increased salt concentration, leading to discomfort and potential cell damage. While this dry eye syndrome afflicts tens or perhaps hundreds of millions of patients worldwide, there are few automated tools to help ophthalmologists with detection, diagnosis, and selection of treatment. The first step in processing these videos is to locate the cornea. Cornea detection has to be robust to variations in lighting, occlusion by eyelids and eyelashes, and rapid movements. It should be fully automated and fast enough to be carried out during a patient's doctor visit. We model the cornea as a circle determined by maximizing the radial intensity gradient as a function of the circle's center and radius. We applied the method to our set of 514 videos, and out of these, only 3 resulted in poor detection results. By far, the slowest step in the detection is the input of the high-resolution video frames. This work relies on numerous well-known Julia packages, and it is now available as an open-source Julia package itself. Using the center of the detected cornea as a reference point, we quantitatively analyze the video. The cornea is divided into overlapping boxes, and quintile pixel intensity data is extracted for each box over the entire video. In this interactive Pluto notebook, we use the collected time series together with information about the box locations to cluster the boxes. By looking at each time series within a cluster, we can figure out which clusters have the rapid and severe thinning that signify breakup of the tear film. This helps us to locate regions of interest for further study. In other work, we intend to explore intra- and inter-patient clustering as well for clinical insights. Tear film breakup is a complicated physical and chemical process that is not well understood. Once we've captured time series of fluorescent intensity over a region of interest, we fit PDE and ODE mathematical models of TBU to the data. By using a reactive Pluto notebook, we have been able to explore the consequences of modeling decisions in real time, and we are now preparing to process the entire data set automatically. This will be an unprecedented new resource for understanding human tear films. In addition to my collaborators and sponsors, I'd like to thank the authors of these Julia packages, as well as the ones I mentioned earlier, and everyone who contributes to the amazing Julia ecosystem.